And now we're going to do the stain. And we're using malachite green, and it's kept in aluminum foil to keep the light out. It's light sensitive. And then we have what I'm going to use in this side tube. On this setup here, I have a hot plate, and I'm boiling the water, and that creates steam, and the steam comes up and it will keep this these slides. So I have on these slides the cells that I've done this the heat fix smear, and I'm going to put a piece of paper towel over them, and then I flood it with the malachite green. And so I have three or so different bacteria on each one of these slides. And so I just drop the malachite green, and the idea is to keep it on here for a good 5 minutes and allow it to soak in, and so the endospores will eventually start to take in the dye. And so then we'll follow up with a counter stain with saffron. And so the difference will be the vegetative cells, or the cells that grow the endospore, will be pink from saffron, and they will also then have the spores stained an emerald green. And then once we've done that, we can look at and see that the spores, if they're in cells, which they may or may not be, if they've been released or not, we can look at their position. If they're in the end of the cell, and these will be rods, so we'll call it terminal if it's near the end of it, or it could be some other place. It could be in the middle of the rod cell, and it is just going to be looked at under the microscope, and then we'll be able to make that determination. And so that's characteristic of a specific genus, is the location of the endospore. And so we're looking just for the presence of endospores is one key point, and then the location within the cell. So this has to steam for 5 minutes, and I'll keep putting on the malachite green until it gets done, and then I'll take it off, and we'll pick it up here in just a few more minutes and do the counter stain with saffron, and then we can look under the microscope. So it's been 5 minutes or more, and it's okay to go more, because the longer you stain, the more the malachite green will penetrate the spore. So I just have to remove this paper towel, and this does stain pretty badly, so you want to make sure you're wearing gloves and things like that. And then I'll take them off. And I just put them on a staining setup. Okay, so now I just have to rinse them off. Now, and you can see that I'm washing both sides, because the stain goes on both sides, and you don't want to get the stain on your microscope very much. So, all right, so I've rinsed off the stain, and now I flood it with saffron, and so the saffron will be the next stain. Do that for a minute. And what that does is counter stain the vegetative cells. And so then that'll give them a pink color, whereas the rest of the cells, rest of the spores will be the malachite green or emerald green color. Sometimes I like to put a puppy pad, it's called the chucks in the hospital, but I like to put it down because this will even stain the surface of your work area, so you want to watch that. Alright, so after 60 seconds, then we can start removing the safranin. And I just put this on a paper towel. And then I'll do my blot dry like I do with a gram stain. Okay, so I just take another paper towel and lay it on it and press down. And go to another area, press down. And remember, you don't rub because that'll remove the cells from the slide. So you don't want that. Alright, now I have my samples. And I can look at it on the microscope, which we'll do next. So this is number 1, E. coli, and what we're seeing is little short rods. They're pink. It doesn't tell us whether it's gram negative or gram positive. We know E. coli is gram negative, but we're looking for the little oval endospores that are emerald green, and they're not there. So that looks like it's going to be negative for spores. This is number 841 Bravo. This is, you can see the rods in the background. They're kind of a grayish, maybe pinkish. Those are the vegetative cells, and then the little pink short rods are actually the spores. So not the pink, but the green rods are the endospores. And so those little green ovals, I call them, those are your endospores. So there's a lot of endospores in this sample. So this is 841 Bravo. Looks to be pretty good for endospores. This is number 3, Bacillus subtilis, and you can see the cells, and then you can also see the green spores, a few of them emerald green. This is number 4, Enterobacter closea, and just the pink rods, no spores. This is number 5, Enterobacter, same thing, no spores. This is Bacillus megaterium, number 6. I don't see any spores that are staying green, although I do see some what looks like spores in the vegetative cells, but so it may be a little bit too young of a culture or some other issue. The staining issue could be. So we'll need to hold a judgment on this one and balance it out with the other data you get for your unknown when you weigh it against this one. So for now, we're going to call this one negative spores. So this is number 7, Micrococcus luteus. Wouldn't expect to see any spores in a coccus, and there are no spores, so that looks fine. This is Sarcina aurantica. I do not see any spores. It's cocci, so again, it makes sense that there's no spores. This is Aromonas sobria. Do not see anything spore-wise. This is number 10, Serratia marcescens D1. I do not see any spores. These are little short rods, but no spores. This is Micrococcus roseus. Again, coccus, or cocci, plural. No spores because it's a sphere. Rhodococcus rhodocrus, number 12. I do not see any spores in this, again as expected because it's a coccus. Alright, so that's all the one. We had a little bit of a question about Bacillus megaterium, number 6. Look that up online and see what if it's a spore former or not. If it is, then just take that into account. But we didn't see any spores in that one.